organisms evolve. That means how organisms change or descendants will change and they adapt over time through changes in their genetic composition. It provides what? The framework for understanding the biodiversity, or only you can say it as the diversity of the life and the relationship between organisms. That means how organisms they form, how organisms are originated, how with change in time, with change in environmental condition, with change in environmental circumstances, how they change themselves, how they origin from one species to another species, origin of one species to another species. And what the theory of evolution explains? It explains the processes like natural selection, mutation, genetic drift, gene flow, recombination, all these are the parts of evolutionary theory. So these all are the basic introduction regarding evolution. Now, according to your syllabus concern, we will first discuss about the evolutionary theory before Darwinism, because we all know that Darwinism is known as the father of evolution due to his contribution to evolutionary biology. And if Darwin or whether any other persons before Darwin they gave any idea about evolution or not, that also we will study in this part. So before Charles Darwin, the concept of species. What is species? If we discuss about the species, that means it is a potentially interbreeding natural population which is reproductively isolated from other such groups. That means they not only look alike physically, but they have some other characteristics. What characteristic they have? An important property mean each of these species, they must be natural species. That means natural occurring species. Secondly, they are reproductively isolated. That means from reproduction point of view, from reproduction structure, from different process of reproduction, they are isolated from other species and they can produce their own progeny for the next generation with any change. So these all are the species, and this concept is known as species concept. So before Charles Darwin, it was considered that species was not changing in accordance with time. But what happened after Darwin, we'll discuss it later. But before that, what was the situation? What was the theory? First come to know that. So Aristotle's Scala Naturae is the 4th century BC. What he proposed? Aristotle proposed great chain of being. That means where all life form could be arranged in a linear hierarchy from simple organism to more complex organism, ones like human. This was not an evolutionary idea, but suggested an organization of life. That means at the time of Aristotle, nobody was given any idea regarding evolution. Aristotle also didn't give any idea regarding evolution, but he just provide one statement that all the organisms are arranged in hierarchical manner. That means simple organism is different from complex organism due to different factors. Okay. Then after Charles Darwin, Carlos Linnaeus, what he stated, he developed taxonomy and classification of living organisms, but believed in 50 of species, fixity of species. That means he didn't give any idea regarding evolution or organic evolution. He just emphasized on taxonomical analysis, how organisms are separated from other organisms, what is the characteristic of organism, what are the fixed characteristic of species. So all these things, all these ideas provided by Carlos Linnaeus. Then George Cuvier, he proposed the idea of catastrophism, where species were created and destroyed by natural disaster. That means It is completely act of God. When species will come, when will when species will disappear from the earth, that depends on natural disaster created by God or any special energy. He was an advocate for species being immutable and opposed evolutionary ideas. That means there is no change in species. Species is constant and there is no evolutionary idea. According to him, there is no evolution. Everything is produced, everything is formed by natural disaster. There is no gradual change or any kind of change happens in environment to the species. So these are basic ideas regarding or before Charles Darwin regarding species and their activities. But after that, one scientist, Jean Baptiste de Lamarck, provided a great idea about evolution. Though the idea was not the accurate idea regarding evolution or that did not provide a great scientific idea regarding evolution, but before Darwin, he was the first. He spoke about the concept of evolution. That means by avoiding others' information, first person was Jean Baptiste de Lamarck, who was famous, a great French naturalist, and he sought a naturalistic explanation for the diversity of modern organism and the animals seen in the fossil record. That means he gave the idea of evolution, but was unable to prove what is the mechanism of evolution. How this organism, he only told that organisms are diversified organisms with descendants, they have some change, but he unable to explain what is the actual cause of that change. That's why he faced a lot of argument. So many people just opposed Lamarck's idea. But whatever may be, he was the first person who gave the idea, who told that there is something known as evolution for which there are different kind of species are present in environment, and the backbone of this diversity is evolution. Okay, so what theory was provided by Lamarck? Just look this presentation. It is known as Lamarckism or theory of inheritance of acquired characteristics. Okay, so it was proposed by Jean Baptiste de Lamarck. What he argued? He argued one species was transferred into another over time. Okay, so for that, just this is the simple explanation of Lamarck. That is, one species was transferred into another over time. For that, he proposed certain explanations. So, what are those explanations? The first explanation is modification of certain organs depending on their use and disuse. Simply, we can say it as use and disuse of organ. So, when there is a necessity of this use and disuse of organ, for that, environmental force is playing a vital role according to Lamarck. Changes produced in environment, the species to adapt the environment by modifying certain organs depending on their use or disuse. If the environmental condition is not accepting the presence of organ, according to Lamarck, the organism just change or separate those organs from their body in next generation. This is the concept provided by Lamarck. That means if there is huge organism, keep the organ with it. If there is no huge, then organism simply remove the organ from the body in its next descendant group. Okay, so What it result? It result the original characters are slowly being replaced by a series of acquired characteristics. Okay. If there is environmental need, organism simply adapt that need by use or disuse of organs. And what after that he told the acquired characteristic. That means if there is a new need, organism try to form a new organ accordingly. If we have three eyes, there is a requirement of another two eyes. Then according to Lamarck, organisms start preparing another two eyes in next generation. That was actually provided by Lamarck, but it was not, he could not prove it. He just explained such kind of explanations. Okay. So what he told, whatever characteristic the organism acquired due to the change in environment, due to the necessity for adaptation, they remain from generation to generation. The used organs present from one generation to another generation, and the misused organs, they simply disappear. Just example we can cite here, the use or disuse of organ. Think about the snakes. According to Lamarck, before or during different evolutionary geological time period or before some days, snakes had legs. By using their legs or limbs, they or reptilians, particularly the snake group, they had legs before some time. By using those legs, they tried to move from one place to another. But what happened? Due to certain nature, due to certain environmental pressure, due to movement or smooth movement in different holes or in narrow passage, they do not use their legs, their limbs for longer period of time. That means there was an environmental pressure. Environmental pressure is for their movement. They do not want to use their legs. And subsequently, what happened? After some generation, the legs were not present in the descendants of those snakes who didn't use their legs. That was the explanation of this characteristic. And the third point explains what? Organisms evolve from simple to complex form because they have tendency towards complexity. Okay, so this is another example we can cite here. Just look at the giraffe. The picture present here, from the beginning to the end, the first giraffe unable to pluck the leaves, the canopy. The second giraffe, in a very difficult manner, it tried and plucked certain amount of leaves. The third giraffe, just average. From the second giraffe, it tried and found more amount of leaves, but in fourth condition, the giraffe able to get all the leaves very easily. So according to Lamarck, this is the environmental pressure. When giraffe population was more and their height was very less, short heighted giraffes were present. The leaf canopy were present at that area at a higher side or high heighted condition. Giraffe tried to elongate its neck generation after generation. So this is the simplest form, and this last one is the complex form. How this simpler to complex form movement is there or conversion is there? Due to its own desire. Giraffe want to elongate its neck, and generation after generation, finally giraffe got the elongated neck for its survivability. That is the tendency towards complexity provided by Darwin. That means use of this neck. Use of this neck means it tried to elongate its neck generation after generation. That is the use of this organ. What I have told in the previous, in the next, in the snake case, misuse of organ. Misuse of organ completely eradicate or disappear those organs from the body. But here, use of organ, that means more power come to that organ. Okay, so this is the idea provided by Lamarck. But this idea was not accepted by many scientists. He might be the first person. For some period, people tried to believe that what Lamarck told was correct. But some scientists during 1890s, some scientists just tried to prove wrong the Lamarckian theory. It was subject to severe criticism. Mainly, it was criticized by Weaver and Weismann at that time, who explains that though the tendency to increase in size has been shown in many forms, there are also instances where there is reduction in size. For example, trees that are primitively larger are large in size, while the shrubs, herbs, and grasses that evolved later are smaller in size. That means, according to evolutionary scientists, what they explain that primitive trees which are formed on earth, they form before the shrubs, the herbs, and grasses. If there is the tendency of complexity, then how a large tree converted to small shrub, a complex form converted to simpler form. So this is the avoidance, this is the rule breaking concept of Lamarckism. That's why they first criticized Lamarckian theory. Secondly, if a new organ were developed in response to new need, then man should have developed wings by now. That means what Lamarck explained, if there is need, organism, any kind of organism, they adapt or they develop those kind of organs according to their need. So human, from many generations, human had a great desire of flying because human unable to fly. If the Lamarckian theory was correct, then till date, human developed its wing, but it is not possible for human to develop wings or to fly. Okay, then changes acquired during the lifetime of organism cannot be inherited by the offspring. For that, if man loses his arm in war or any kind of accident that severely affect the man and he loses its eye, the next generation will not be blind. Okay, if Lamarckian theory was correct, then next generation to generation, the man or the next offspring groups were completely blind. But that is not happened in general sense. That's why Lamarckian theory was opposed by Weismann and Quiver at that time. Weismann also emphasized on somatic changes acquired during the lifetime of one is somatic, another is germinal. Somatic means physical, germinal is the inherited gene bearing quality. Somatic changes acquired during the lifetime of organisms are non heritable, whereas changes in germplasm or reproductive cells are inheritable by offsprings. So, what Weismann told, he told that Lamarckian concept was partially correct, but what Lamarck told was not correct at that time because, according to Lamarck, whatever change occurred in somatic characteristic, that it changes in next generation or that will inherit to the next generation. But Weisman proved it wrong by providing many examples. And what he explained that if change, any kind of change happened in somatic characteristic, that never transmit to the next generation. Only the change in germplasm characteristic or reproductive cells of the organism, that only transfer to the next generation, because the gametes only transfer from the parents to offspring in next generation. That's why any kind of change in gamete property only change to the next generation. That is the Lamarckism and criticism to Lamarckism. Now, neo-Lamarckism was provided by many scientists. What it explains, it is a modification of original theory of Lamarck in order to make it more suitable to modern knowledge. Neo-Lamarckism does not give any importance to the factors. The theory stresses on direct effect of changed environment on the organism, and normally only those modifications are transferred to next generation which influence germ cells or where somatic cells give rise to germ cells. Okay, so these things already we have explained. The fourth point is important point. That means if any change occurred in next generation, for that change, germ plasms or reproductive cells are important. Reproductive cells are playing important role, not the somatic cell. If the reproductive cells are affected or the reproductive cell affects the somatic cell, then only, somatic cell affects the reproductive cell, then only reproductive cell change, transfer those chains to the next generation. That already we have explained. Okay, Charles Lyell, another scientist, another geologist who also worked on uniformitarianism, that is suggesting that the earth was shaped by gradual processes over long period, strongly influenced Darwin's idea about slow and gradual evolutionary change. We'll discuss about it later. Darwin's idea about slow and gradual change, gradual evolutionary change. Okay. So up to this, is it visible, audible to all?
Yes, sir.
I think you got some ideas, a little bit idea about Darwinism, criticism to Darwinism, Lamarckism, criticism to Lamarckism, and neo-Lamarckism. This is the thing happened pre-Darwin period. Okay, but evolutionary concept changed during Darwin, Charles Robert Darwin. Okay, because he provided certain scientific theory for the process of evolution. Though Darwin's all theory were not accepted by the evolutionary scientists in next time, but many theories or the modern theories based on Darwin's theory for evolutionary research. Hence, he was considered as father of evolution. So he actually revolutionized the concept of evolution with his theory of natural selection outlined in his book Origin of Species. That means how species are formed. What is the origin of species? Actually, provided the idea provided by Charles Robert Darwin. Actually, Lamarck was the first to talk about evolution, but current knowledge of genetics demolished his theory. Since acquired characters such as muscle development are not inheritable after development of genetic theory, only those characters those whose information resides in the gene are inherited. The theory of evolution or Darwinism was not developed solely by Darwin because another scientist also known as Alfred Russel Wallace provided the idea which was almost similar to Darwinian idea. That's why many people accept Darwin's idea because at that time Wallace and Darwin were provided the similar kind of idea which supports the evolutionary theory. Darwin, for this evolutionary research, he made a 5 year trip around the world. Okay, all we know that the voyage started from England to North America and different part of the earth. Then they return back to their base after 5 years. During this 5 year trip, he collected the information, not small group of information, a lot of information were collected by Darwin. And when he accumulated all the information, when he analyzed all the information in his laboratory, he got astonished, he got abashed. And after that, he just explained all the explanations, all the informations in his book, The Origin of Species, which in latter provided many ideas about the theory of evolution. Okay, what Darwin explained in his theory, Darwinism defends the evolution does not depend on environment change, but on genetic randomness. According to Lamarck, evolution was due to environment, but Darwin explained that directly environment cannot affect the organismal evolution. For organismal evolution, genetic randomness, that randomness might be affected, might not be affected due to environment, but genetic randomness is actually the source of evolution at that time. So, in his theory, he explained five important factors for the process of evolution, and particularly he emphasized on natural selection. Okay, so what explanations provided by Darwin? He explained first, variations is the property of each individual. That means individual within a species exhibit variations in trait. No individuals are similar, same with other individual. There may be similarities, but no individuals are same with other individuals in the species group, in the same species group also. Heritability is the variations are heritable and passed down to offspring. If any kind of variations are found in any species and that variation is present in germ plasm or reproductive cell, that is passed to the next generation from parent to offspring. The next one is differential survival and reproduction. What differential survival and reproduction explains? That individuals with traits better suited to their environment are more likely to survive and reproduce. It is actually the concept of natural selection. That means survival of the fittest. Every organism try to produce more and more number of organisms, but it is not possible for all the organism to survive because resources are limited. So for those limited resources, organismal fighting for resources or competition among the organism for resources generally found in the environment. So who will survive? Who own the competition, who is able to get the resources in proper amount after the competition. That is only the species which can survive. And according to Darwin, The species was known as fittest species. The fit one will only survive, not others. When the fit one will survive, the fit one can reproduce the next generation with these fittest properties. That means the organism which can survive, it was the fit organism, because organism bear the fittest gene, and particularly those genes are transferred, inherited to the next generation from parents to offsprings. Okay, so natural selection was the famous theory of Darwin. It stated that evolutionary changes come through the production of variation in each generation and differential survival of individuals with different combinations of these variable characters. Okay. Darwin emphasized on gradualism because this change is not a rapid change, a speed change, a quick change. He told that this is a gradual change. That means generation after generation, certain fit characteristics adapted by the organism, and those characteristics are accumulated in its own cells, which is transferred from one to next or parent to offspring. Impacts of Darwin's theory. Darwin's work provided a mechanistic explanation for how species change over time and challenged the long expanding belief that species were fixed. That means individuals with characteristics which increase their probability of survival will have more opportunity to reproduce. That is the survival of the fittest. That means the species which can survive, they are considered as fit, they can produce their next generation. Okay. So this all about Darwin's idea and Darwin's explanation about natural selection. This is an example of natural selection we can cite. The industrialization, before and after industrialization. You can mark here, before industrialization in generation 0, what happened? Biston betullaria was the species. The dark and grey color moths were present in England. Before industrialization, the tree trunks were white because there was no soot deposited on the tree trunk. Whatever lichens present on the tree trunk were white in color. That's why these white color species were present more, because the dark one is completely visualized by the predator. Whatever predators, birds are there, they can visualize easily the dark one because of due to this contract color. Okay, so dark population before industrialization was less. Why? Nature select the white population in comparison to dark population. What was the nature? The tree trunk where they were present, they were attached. Okay, but what happened after industrialization? After industrialization, so many pollutions, so many black dust, soot, everything deposited on the trunk of the tree. But that is the change in environment, that is the environmental pressure. What pressure exert on the species group? Just mark this picture. The trunk was completely suitable for the color of this black moth. They can develop a good quality of camouflaging on this tree trunk. But the white color moth easily visualized by the predators, and predator can easily capture the moth for its food. This is the actual picture. on the trunk. This is the comparison. Just look at here. On white surface, the gray marks unable to visualize due to its camouflage nature. But on white color surface, the black marks were clearly visible. What happened in the previous diagram. And the opposite thing happens here. The black one, it is difficult to detect the black one due to camouflage, but this one clearly visible. So this is the example of natural selection provided by Darwinism. This situation is the post-Darwinian evolutionary thought, means after Darwin, how Darwin's theory for many years, most of the people, they accepted Darwin and what Darwin explained, accepted by many people of the world, but that theory was opposed later due to some other research, other discovery of factors. One scientist known as Gregor Johann Mendel, all we know that he is well known as the father of genetics. Discovery of genes, genetic factor, played an important role in evolutionary biology. While Darwin's work lacked a clear understanding of inheritance, Mendel's experiment with pea plants provided the foundation for genetics. Darwin only told that some factors are there which transmit from one generation to another generation, but unable to explain what was the factor. And that factor was explained by Darwin and his co-workers in next period of time. Okay, Mendel's work was discovered in 20th century. Before that, Darwinian idea. Mendel's work was actually before Darwin's idea, but work was discovered, work was analyzed in early 20th century. Darwin had already explained his idea, leading to the integration of genetics with evolutionary theory. So based on that property, modern synthesis played an important role between 1930s and 1940s. This period saw the merging of Darwinian natural selection with Mendelian genetics that combinedly explain the modern synthesis, the modern evolution. It leads to what? Leads to what is now known as modern synthesis. The key figure like Ronald Fisher, J.B.S. Halden, Sewall Wright, they contributed to development population genetics, which quantifies how evolutionary processes like natural selection, genetic drift, mutation, and gene flow affect genetic variation. Okay, so these are the scientists who had played a great role for this modern synthesis. And what modern synthesis actually emphasized, three important points I have cited here from modern synthesis. The first one is evolution is gradual and result from small genetic changes. Evolution is not a great large thing. It's small changes which are gradual and gradually accumulated in generation after generation. Natural selection acts on genetic variation. If genetic variations are there, nature will select always the fit variant, not the weaker variant. So natural selection is also playing an important role. It means the fit variant adapt the changing environmental condition, not the weaker variant. Whatever pressure is provided by the nature, provided by the environment, that can only be bear by the fit variant. Speciation occurs as population become reproductively isolated. Sexual reproduction is another important factor for this evolution. That means when more different kinds of individuals they come together. That means if we are talking about the male and female, male had certain other properties, female had certain other properties. Twin combination occurs. Some male gene and female gene they exchange and can form a new gene. Okay, that is the species formation, speciation characteristic. Okay, so these three are important properties provided by modern synthesis. Modern synthesis, again, I am saying modern synthesis is the combination of Darwin's idea and Mendel's genetic description. Okay, so for this post synthesis development, molecular evolution, natural theory of molecular evolution, everything was come to light. The modern evolution explained about DNA sequencing, natural theory of molecular evolution explained about uh, most evolutionary changes at the molecular level are due to genetic drift. We'll discuss it later what genetic drift is, what natural neutral mutation is. Okay, provided by Moto Kimura and evolutionary developmental biology, EvoDevo investigated how changes in gene expression during development contribute to environment evolutionary changes. Everything was the ideas after the post synthesis or modern synthesis. Okay, so Darwinism at certain point was criticized by these scientists Fisher, Haldane, Wright, Dobzhansky, Mayer, Huxley, Simpson, Stebbins, Richard Dawkins, Fukuyama, Gould, and Smokovic. Okay, so what they explained was the another important theory for evolution, and this theory till date was accepted theory. That this theory based on Darwinism, that's why this is known as neo-Darwinism. That means sudden modification or certain modification with Darwinism is known as neo-Darwinism. And for this neo-Darwinism theory, these scientists they played a great role for this theory formation. What this theory explained, this theory does not accept the inheritance of acquired characteristic. That means the Lamarckian theory. Because Mendelian genetic contributions are there. According to Mendel, the change which is found in the next generation, in the offsprings, is possible due to the change in gene. But that was not explained by Lamarck. That's why Lamarckian theory was proved wrong at that time because Lamarck focused on somatic characteristics. But genes are not transferred from one generation to another generation from the somatic cells. Next is in sexual individuals, the only source of genetic variability are mutations. The idea of mutation provided by theory of mutation was provided by Hugo de Vrij. What it explains in the individual with sexual reproduction, mutation, genetic recombination, and natural selection are responsible for genetic variability. Mutation means any sudden change in genetic characteristic due to any factor. That idea was provided by Hugo de Vries, and in later stage, this mutation idea actually plays an important role in evolutionary concepts. Mutation is the only cause for evolution. Okay, detail of mutation we'll discuss about it in later. The population evolves, not the individual, since the result of reproduction is what leads to evolution contribution of population genetics. That means if change occurs, it is not meant for a change in the particular species. Change in the population means there is evolution. And according to them, evolution occurs gradually. In this point, they accepted Darwin's concept. Evolution is the result of small changes in the allelic frequency, what is known as population frequencies. For that reason, having a new species is very long process, contribution of population genetics. Evolution is not a speed process, a faster process. It's a slow process and it will take a long period of time. The speciation occurs when they arise reproductive isolating mechanism. That means speciation means completely new species are formed when there is reproductive isolation. That means from previous group, it is reproductively isolated, completely reproductively isolated. Then only you can say it as there is speciation. Okay, so this all idea about neo Darwinism. So this all about the evolutionary introductory idea. I think, is it audible? Am I audible to all?
Yes, sir.
Yes,
Yes,
sir.
sir.
Okay. Now we'll go for the next part. I'm just sharing the next slide. Just wait for 2 minutes. Okay. Now we'll discuss about evolution and molecular biology and new synthesis. In Darwin, post-Darwinian period, many scientists gave some idea about the new synthesis, how speciation occurs, how organisms are getting diversed from one group to another, how new species were formed. Okay, that basically explains about mechanism of evolution. Okay, so for evolution, molecular explanation is actually the explanation for evolution in comparison to other explanation, because molecular explanation easily accepted by many people, many scientists. Okay, so the modern synthesis of evolutionary biology and molecular biology has reshaped our understanding of processes of evolution at molecular level. Whatever idea provided during Darwinian period and pre-Darwinian period, so many ideas are reshaped the thinking, the understanding of evolutionary theory. So what actually it emphasized on four important things. Just look at this blue marks things: molecular evolution, genetic code, molecular phylogenetics, gene duplication, and divergence. These are very important concepts. Okay. So first of all, we'll go for molecular evolution. And under molecular evolution, what were the theories for this molecular evolution? This process by which genetic material changes over time, but this change are driven by certain processes. What were the processes? Mutation, genetic drift, natural selection, and gene flow. Out of these four, these are mechanism of evolution. And out of these four, already we have discussed about natural selection in the previous slide. That's why all these rest three things we discuss in this slide. Okay, so first of all, we'll briefly discuss about mutation. What is mutation and how it works on the process of evolution. That is the first idea we'll discuss about or regarding this molecular evolution. Okay, so what is that mutation? It is the change in DNA sequence of an organism or any kind of small or larger group of organisms. If there is any change in DNA sequence, genetic properties, then we can call it as a mutation. What are the causes of mutation? It is caused by errors during cell division or by exposure to DNA damaging agents in the environment. Okay, such as chemicals and radiations. We know that the common mutation causing factor is the X-ray. Not only X-ray, is the UV ray, but not only UV ray, many other factors, chemical factor, radiant factors, they are also responsible for mutation. Mutation means change in the genetic property. Just look at this picture at the right side, the first one, which I am just showing my cursor. This is the DNA sequence. That means these are the base pair arrangement where the curve arrow mark is there. These are the base pair arrangement. Just look at here. This is the purple color, and under this is the green color. Okay, this is the original property. If we look the correct copy, the original copy, this looked like this. Purple color, and one strand is there, another strand is here. Okay, this codon bears this purple color and this one the green color. But what happened if mutation occurs? This copy is known as the mutant copy. That means the correct copy, correct original copy is changing to mutant original copy. Why? Because this change, this particular structure is getting changed to another structure here. Okay, so this is the concept of mutation. Mutation means any error happened to the genetic constituency of the DNA. DNA genetic constituency is changed due to any kind of chemicals or radiations that leads to mutation. Okay, so this might be the environmental pressure. This radiation is from environment. Certain chemicals from environment also they cause the mutation in the species. So mutations are harmful mutations, beneficial mutation, or having no effect. That means all the mutations are not dreaded mutation. That means they, in all circumstances, they are not responsible for the death of organism. Those mutations which are responsible for the death of organism. If we are talking about the skin cancer, which is caused by UV rays, in that case, we can call it as the mutation is harmful mutation or lethal mutation, which leads to death of the organism. But in general sense, all the mutations are not for organismal death. Certain mutations are also called as good mutations, and those mutations, good mutations, helpful for organismal survivability in dynamic environments. Okay, and those mutations actually happen in the genetic level of organism, which can pass from one generation to next generation. That's why the mutation property, the mutated property, is also present in subsequent generation, which help those organisms to survive in changing environmental condition. Okay, so these are important aspects regarding mutation. So many mutations are there. I have cited here certain mutation due to stipulated time, substitution mutation, deletion mutation, insertion mutation, inversion mutation. In each case, you can mark here these pictures. Substitution means the substitutes are there, not the previous things are present. The substitutes are present. The substitutes. These are the substitutes which substitute the original substances. Deletion, that means eradication. Eradication means whatever structure was present, not maintained in future. Insertion, addition of one base pair. Inversion, just reverse of this base pair. So these are many kinds of mutation. Point mutation is there, which replaces one nucleotide sequence with another. Substitution mutation is there, silent mutation is there. So different kinds of mutations are there. So basically, this mutation is considered as the backbone because mutation bring changes in genetic level. Without mutation, there is no change in genetic level. That's why mutation is considered as Hugo de Vries' discovery. This mutation is considered as the backbone for the evolution. The second factor is genetic drift from this molecular evolution. What genetic drift explains? It explains about random change. It is also called as random genetic drift. That means random change in gene frequency. Gene frequency, what actually the gene frequency or allelic frequency? The characters, the genetic characters. Alleles are the characters. Genes are expressed by alleles. One is dominant allele, another is recessive allele. When alleles are expressed in a population, or how many times that alleles are repeated in a population, that determines the frequency of alleles. Okay, one is allelic frequency, another is genotypic frequency. Genotypic frequency explains what? It explains how many individuals are present. That frequency is known as genotypic frequency. So allelic frequency at the gene frequency. That means the genes are actually expressed by allele characteristics, dominant and recessive allele characteristic. So how many times those alleles are expressed? Because all the phenotypic characteristic are the expression of genotypic characteristic, and these genotypic characteristics are maintained by dominant and recessive alleles. So how many times those characteristics are repeated in the population group is the frequency of those alleles. What genetic drift explains? If the frequency is continuous, one generation to another generation, that means there is no evolution. But if the frequency is changed due to by chance property, that means no nature design property is there. By chance, any kind of substances, the frequency of alleles or variant from one gene changes by chance. This is known as genetic drift. Okay, just look at this picture. how genetic drift is explained here. Just look at this population of beetles. Here, the examples are given red and the first one, this one, where my cursor is there moving. The first picture showed that beetle red and blue both are almost equal in number. Okay, but any factor is there, environmental factor. Here is a capture net for the beetles, which captured most of the blue beetles from the population. Before it, the population constituted 5 number of red and 5 number of blue beetles. But in the by chance factor, that means there was no pre-decide factor. By chance, this thing come to their population and 4 number of blue were completely eradicated from those population, were completely vanished from those population. That's why the ratio become 5 is to 1 in position of 5 is to 5. What happened in next generation? Only the red population, they were present more in number. The primary population was equal. That means 5 blue, 5 red. But here the last population, the ultimate population, they bears only two species number of blue, but 10 species number of these red. That's why this kind of change is known as genetic drift change, which is actually explained by these two effects. One is bottleneck effect, another is founder effect. That means if certain species are present in one population, and from this, just look at this total population. It contains yellow and red, dark red species. But some species, they migrate to other area due to any kind of factor, due to the factor of reproduction, due to the factor of protection, due to the factor of nutrition, due to any kind of factor, they shifted from one area, from their original location to the new location, and they reside there in the new location. They improve their population, they develop their population, they do not come to the previous location again. That means those species, they migrate and establish their population there, and they try to form new species. In new species, what will happen? More number of yellow populations in comparison to red or dark red population, or it leads to extinction of dark red population in the next generation, because in the new area only one species is there, no chance of reproduction. That's why that species is responsible, or that species might have destroyed after some days or times. This kind of effect is known as founder effect, and those species where first established a new population are known as the founder species. This happens in natural ecosystem also. Here explanation is given for you. Founder effect. The new population is founded in a new location due to physical or geographical barrier. The new population form does not interact and mate with the original population. As a result, the allelic frequencies of new population will be different from the original population. Just look at this picture. At this condition, the allelic frequency is the allelic frequency in this total population. But for new population, these allelic frequency are the total allelic frequency. That's why there is change in allelic frequency from one to another population, which leads to evolution. What bottleneck effect explains? Bottleneck effect explains the population size severely decreases due to competition, predators, or disease. The frequency of certain alleles in a population changes because the organisms that carry them are eliminated. The others increase in number because they are the only alleles left. Just look at this picture. If this was the parent population, any kind of natural disaster or due to any kind of natural disaster, loss of many parent population happened. And some of the population they left, which can pass, which can easily protect themselves, or which can struggle, and they survived during that phase. Just look at this picture previously. The blue and yellow were almost same, but in the next generation, that means that maintains a particular allelic frequencies. But in next generation, in the new generation, you can find that more number of blue species are there in comparison to yellow species. What it result? It result change in allelic frequency from this previous one, from the first one, from the original parent population. That's why we can say it as there is evolution occurs. And these evolutions are actually represented by the factor of evolution were known by a mathematical calculation which was commonly known as Hardy-Weinberg equation. We all know that the frequency of dominant allele plus the frequency of recessive allele is always 1. If we express it in genotypic format, frequency of homozygous dominant plus frequency of heterozygous plus frequency of homozygous recessive is always 1. That means p square plus 2pq plus q square is always 1. If population is maintaining 1 in any or generation after generation, that means there is no evolution. But if the value shifts from 1, there is evolution, definite evolution. Okay, so this is all about genetic drift. Now the factor is called as the gene flow factor. What gene flow explains? Or gene flow explains movement of genetic material. Just look at this picture. Here, one population of red beetles. Another area, the population was yellow beetles. Generation after generation, red beetles do not pass to the yellow beetles. That's why their population maintained its genetic integrity. But when, due to any factor, due to any possibility, if one or two or group of one population will migrate to another population, that means gene migration occurs at that sense. And this gene migration is responsible for changing the allelic frequency or genotypic frequency. Genotypic or allelic frequency means how many individual alleles are there, how many total alleles are there. Individual alleles divided by total alleles is known as the frequency. So here, whatever total alleles are there, they were represented by yellow beetles. But when the red beetle come in the group of this yellow, that means the frequency is definitely changed due to this migration. So if there is change in frequency, there is change in Hardy-Weinberg principle, law. That's why you can say it as there is migration or there is evolution. So gene flow is the expression of this gene migration. Okay. It can occur when individual migrate and reproduce in new population. That is the gene migration. Just look at the picture below. Two groups of populations are there. If the population exchange occurs, they can affect the frequency of the original population. That is the effect of this gene flow. It is important because it maintains genetic variation. If this particular group of organism, the yellow population, they unable to protect themselves from a particular disease, but this red population, they protect themselves from a particular disease. When the red come in contact with yellow and they mate with yellow, producing a new category of species, the species having the properties of this red and yellow can survive, can tolerate the disease, and can produce same kind of organism in next generation. That's why it maintains the genetic variation. Okay, so these are certain importance of this gene flow. Gene flow is affected if there is barriers, if the nature will not allow the organism to pass from one to another, if the two populations are far away from each other, then this gene flow will not occur. Okay, so this is all about the gene flow, and natural selection already we have explained. So this all about the properties of this molecular evolution. First thing we have covered. The second one is genetic code. We all know that the code is very essential because they code for certain amino acids, which are the building blocks of proteins. Proteins are the building blocks of our body, the cells. So genetic code can be defined as set of certain rules using which the living cells translate the information encoded within genetic material. That means how our DNA or RNA are formed depends on this code. If there is change in this code, then we can say it as there is mutation. Okay. So the ribosomes are responsible to accomplish the process of translation. We all know that the central dogma theory, DNA to RNA, RNA to protein, the central dogma theory. If the RNA can arrange these codes in proper format, then normal organism or normal production is possible. If there is change, that means that leads to change in the species property. If the change is good, not harmful, then that leads to the evolution of organism. That is the relationship of this genetic code to the evolutionary process. The genetic code actually link amino acids in mRNA order using molecules to carry amino acid and to read mRNA 3 nucleotide at a time, like phenylalanine, leucine. Phenylalanine is expressed by triple U, UUC. Leucine is expressed by UUA, UUG. Okay, so all these are the different letters. By using those letters by triplet codon format, the picture shown here, just look at this picture how RNA is formed by this codon. Codon 1, codon 2, codon means the genetic codon. GCU, ACG, GAG, all these are the genetic codes. These codes represent the amino acid structure. Now, the next important is molecular phylogeny. What phylogeny explains? It is the study of evolutionary relationship among biological entities or individuals. Not only individuals, populations, species, Our taxa also included in this group by using a combination of molecular data such as DNA and protein sequences, presence or absence of transposable elements, and gene order data and the statistical technique. Just look at this picture. This was first developed by Fitch and Margolis in 1967. Just look at this picture. What is molecular phylogenetics? In molecular phylogenetics, the study of evolutionary relationship. That means, just look at here. Here, crocodile, birds are there, lizards, snakes are there, rodents, primates, and marsupials are there, and they originated from common ancestors here. Again, those common ancestors are super common ancestors. That means a single group of organisms are diversified into many groups. This diversification is possible due to evolutionary process. So by comparing this evolutionary tree structure, we can be sure that which organism is evolved from which organism, or which one is the common ancestors for which organisms. Okay, so these are actually the presentations of molecular phylogenetics. Molecular phylogenetics is another important branch by which we can get idea about the molecular evolution. Then the next one, the last one, is gene duplication and divergence. Gene duplication express what? It expresses the process that increases the number of copies of gene or chromosome, which can help cells make protein with multiple function. Just look at this picture. Ancestral state gene is there. Duplication occurs. Different duplications are responsible for different production of proteins. Divergence, what explains? It's a process that causes genetic differences between group to develop. That means where we get actually the diversified, that means if this is the common ancestor, certain changes in genetic groups are there. Duplication leads to formation of many protein factors. But if there is any change, if there is any divergence, that leads to different characteristics. Some characteristics are maintained or changed to another properties. From a common ancestor, birds and crocodiles were developed. That is due to gene duplication and gene divergence. Paralog is another important concept which is dependent upon this gene duplication. That is the duplicated copies of gene that result from gene duplication. Paralogs with similar sequences and function from gene families. These are, that means the duplicated property from one family are explained as paralogs. Another important factor is evolution of new enzyme. If gene duplication and diversification occurs, genes are responsible for the production of these enzymes. Mostly the enzymes are proteins. So if there is change in properties, protein properties, there is change in enzymes, the catalysts. If enzymes are changed, diversified groups are formed. That leads to evolution. So these all are the factors, the mechanisms for evolution. Then the origin of life from the molecular point of view. Many theories are there for the origin of life, how life originated. We all know that Earth originated, and this theory was explained by Big Bang theory. The origin of universe, the origin of solar system, the origin of earth. But when the earth was formed, it was not suited for the life, or it unable to support the life form. After so many days, after certain important changes, life was organized on earth. But how? Life do not form from the life. Very first life formed from the abiogenesis. The theory of abiogenesis was the first important theory for this origin of life. How? Just look at this picture. Whatever condition were present at primitive earth was the reducing condition. Okay. Before that, certain theories, before this is the theory, scientific theory, but before that, spontaneous generation theory, special creation theory. Spontaneous generation theory explains spontaneous development, spontaneous formation of certain category of species. Special creation explained everybody is the contribution of God, no other for the organism. Cosmozoic theory, it explains the evolution of species, the formation of species from the cosmos. Panspermia explains from the celestial body, spores or spores come to the earth and they develop into new life. But when chemosynthesis theory was developed, and particularly the concept provided by Oparin and Halden, they actually proved, they actually explained the idea, but they unable to prove their idea. In latter phase, the idea were proved by Miller and Urey. In next slide, we'll discuss about it. But what Oparin and Halden explained, they stated the chemical evolution of life. According to them, the primitive earth was devoid of life due to reducing environment. No free atmospheric oxygen was present. Free atoms are present. No free oxygens are present. Look at here. Carbon, hydrogen, oxygen, and nitrogen at its atomic stage was present. But organism unable to use the atomic oxygen. Organism can use the free oxygen, the molecular oxygen, which was not present in previous atmosphere. Okay. So what happens? Abiotic synthesis of monomers, that means combination of these simple inorganic substances, abiogenesis, that means the substances they didn't have life, simple substances combined to form this certain intermediate complex substances. Hydrogen combined, hydrogen atom to molecule, hydrogen to oxygen, they form the water, ammonia, methane. Then amino acids, purines, pyrimidines, monosaccharides, fatty acid, glycerols, they are formed. When they united, they form monomers, combine to form polymers. Okay. Then proteins, nucleic acid, fat, polysaccharide, which are the main cell building materials, they were formed. Then formation of coacervate, cell-like substances, but not cells. Then formation of protocells by accumulation of biomolecules, and at last origin of this protocell when they developed this kind of substances like nucleic acid arrangement and protein controlled metabolism, that means the presence of catalyst. Okay, the protocells were formed, and these protocells very rapidly they grow and they form what different domains of life: bacteria, archaea, eukarya, which we'll discuss in next unit. The three domains of life. LUCA. Last um, universal common ancestry. Last universal common ancestry. And they in further they develop different life forms. We'll discuss about it later. But this was the property, this was the concept provided by Oparin and Halden, how primitive earth is responsible for formation of life. That means life was born from abiotic for or combination to the biotic combination. Okay, that was the idea provided by Oparin and Halden. That is the abiogenesis. Life began from non-living molecules through chemical evolution, leading to the formation of simple organic molecules that eventually gave rise to more complex structures like RNA and proteins, the nucleic acid structures. Then prebiotic chemistry explains what? Earth's early environment contained molecules like water, methane, ammonia, hydrogen under the right condition. These are actually the energies, lightning, volcanic activities. The molecules could react to form. How these molecules were stable at that time, but when these lightning, volcanic eruptions occurs, they were responsible in combination of molecules and formation of new complex substances, which in further develop to formation of life. Then protocells, simple membrane bound vesicles that they form protocells. These are not actually the cells. They are protocells. But protocells, when start dividing due to presence of this nucleic acid structure, the genetic material, they converted to cell. And in further, they developed to three domains of life: archaea, bacteria, and eukarya. So life originated from RNA, which is called as RNA world hypothesis. It was supported by certain evidences. In next slide, we'll discuss about this RNA world hypothesis. But evidences where we have now ribozymes, which explains the catalytic property. Certain bacteria, eukarya, also they have ribozymes. That means it provide an evidence that previous organisms also they have ribozymes, which played a vital role in catalytic activity. Okay, catalysis. Self-replication property, some ribozymes have been shown to catalyze their own replication. What it suggests? It suggests that early life could have relied on RNA to simple replicate and evolve. They do not rely on DNA because DNA is the complex structure. RNA is the simpler structure. That's why RNA as a precursor to this DNA. RNA likely preceded the DNA. That means RNA was first formed. The simpler substances always form first in comparison to complex. This is the great evidence. RNA was the simplest substances in comparison to DNA. And RNA was unstable, DNA was stable. So first when formed, it was unstable. Then it converted to stable substances from RNA to DNA. RNA in modern biology, even in present day life, RNA plays crucial role. Okay, supporting idea. That means DNA, they form first RNA and RNA form different types of protein in central dogma theory. That's why RNA from the previous cell to the modern cell, they bears the important property. Okay. Now introns. Introns are another important aspect of this chapter. We have to discuss about these introns. What actually these introns, we have to first get some idea about those introns. Introns are actually the uh, non-coding segments of the DNA. The DNA structure, the non-coding structure or DNA of DNA or RNA within a genetic gene that is removed before the RNA is transferred into protein. So where it actually found the introns, just look at this picture. They found most in eukaryotic genes and eukaryotic variants. Certain exons are there. They exons are connected. The non-coding segments, these are the coding segments, these are the non-coding segments present. Introns are present between the exons. Basic function of this, they allow eukaryotic cell to evolve new genes. Okay, splicing and all these things are very long concept. No time to describe. Just have some idea about this. What is intron? They are non-coding sequence within the genes that are removed during RNA splice. Okay, what are the key points for these introns? Introns are believed to have been present in earliest common ancestor of eukaryotes. Exons actually responsible for different properties, but in previous life they do not show different properties. Many lives they show common properties. That's why no variations are there. That's why it was believed that introns are present in previous life's genetic material. The intron early hypothesis explain or suggest that they existed in earliest gene and may have played a role in facilitating the recombination and evolution of new proteins. Introns may have enabled exons, shuffling a process that accelerates the evolution for new proteins by mixing and matching functional domains. So, what are the role of these introns in evolution? They may have played a role in increasing genetic diversity. Due to presence of this intron, we actually got this genetic diversity, the variance and complexity by allowing alternating splicing. How many splicing are there? That determines the variant or variations in genetic property. More variation, more evolutions. Splicing mechanism actually is responsible for intron removal, lysosome, which is ancient and complex, suggesting that introns and the process of splicing have deep evolutionary roots. Okay, transposable elements also. Some introns derived from transposable elements, DNA sequences that can move within the genome, which have contributed to genome evolution by increasing variability. Already I have told you, the more variation, more genetic variation, more evolution. Introns in the previous life played an important role, or in ancient components of genes, they played an important role for the variation, which in turn leads to process of evolution. Okay, so this all about the introns and their importance. And here another important factor, important aspect we all have to understand, that is RNA world. Just little bit more we will discuss about this RNA world. I'm just sharing another slide to you. This is the RNA world hypothesis. Already we have explained that RNA was the first genetic material on early life. Early life on Earth was based on RNA, ribonucleic acid, rather than DNA and proteins. Because proteins are formed from DNA, sorry, proteins are formed from RNA, and DNA in latter phase is the complex structure. It is formed after RNA. So RNA was hypothesized as the first genetic material in early life. This background already we discussed in the previous slide. No need for its discussion. Then why? Question is why RNA? Why we're talking about RNA? Because it's a simpler structure than both DNA and proteins. Already we have discussed. It can act both as replicating molecules and catalytes, which could have helped jump start life before. That means when life was started, it were more simple things. A single thing can perform many actions. If we are talking about a single cell, unicellular organism, that means a single cell is performing all the kind of actions. But in multicellular organism, different cells are responsible. There is division of labor. That's why if we are talking about the replicating property, if we are talking about the catalytic activity, nowadays the replicating property is performed by genetic material, DNA, RNA. The catalytic activities are performed by enzymes. Okay, but previously, in previous format, in previous life, earliest life, they didn't have certain different organs, different products in their body for different action. A single product can perform a many function or single product have many functional ability. That's why it was thought that RNA was the first genetic material in previous life. Ribozymes as catalysts, the discovery of ribozymes in 1980s provided key support for the RNA world hypothesis because ribozymes are RNA molecules with enzymatic activity, demonstrating that RNA can function as both genetic material and catalyst. The self-replication property also the beer. Just look at this picture here. Primitive atmosphere, amino acid, nucleotides, they're simple substances. Okay, they form this RNA. After that, DNA was formed. But RNA, when combined with certain other properties, membrane developed on it that responsible for formation of this protocell. And finally, from this protocell, different cell structure, prokaryotic and eukaryotic cell structures were evolved. Okay, so for this region, RNA was considered was the first nucleotide, nucleic acid material, first genetic material in the body of very simple organism, protocells. This picture is showing the RNA wall hypothesis. RNA from inorganic sources, they form RNA self-replication property was there. That's why from one RNA to many RNAs form. RNA catalyzes protein synthesis at that time when body needs its internal energy. Catalytic activity is playing a very important role. And for that, RNA is playing important role. Okay, the dual role is the main important property of this RNA. Genetic role is RNA like DNA can store. It means nowadays DNA is the main genetic material storing factor or transmit genetic information, making it a good candidate for the first hereditary molecule. RNA was considered at that time was the first hereditary molecule because no DNA was found in the first cells, protocells or first cells on Earth. It has catalytic role at that time because some RNA molecules known as ribozymes, already we have discussed, the catalyzes chemical reactions. At that time, when chemosynthesis occurs without any photosynthesis organisms were survived, these ribozymes were played important role. Okay, so these are different abilities of RNA for which we can assume that or we are assuming that RNA was the first important genetic material at that time. Prebiotic chemistry explains abiotic synthesis of RNA. RNA world hypothesis fits into broader context of prebiotic chemistry, the study of how organic molecules would have formed from inorganic precursor. Okay, the Miller-Urey experiment actually explained the Oparin-Haldane concept. They demonstrated that simple organic molecules can be synthesized under the condition that simulate early Earth's atmosphere. Just look at this picture here. This picture. Look at this picture. Miller-Urey experiment. What they tried to signify. The experiment actually explains the presence of early atmosphere. Early atmosphere contain these type of substances. They take those substances which were explained by Oparin and Haldane. Previously, we have discussed about it. Oparin and Haldane explained the previous atmosphere contains this methane, ammonia, water vapor, hydrogen gas. So, for this presence, they took in a vessel. This is the vessel. In that closed vessel, they took this ammonia, methane, and hydrogen in their ratio, appropriate ratio. And for adding water vapor, they just make a boiling water, and from this part, more and more amount of water vapor are added to this part. Okay, and for their energy, for their combination, energy is required. In the previous atmosphere, energy was provided by sun, energy was provided by volcanic eruptions, energy was provided by different lightning activities. So for that energy, they use this electrode. When the sparkles, the spark discharge, that make the combination of these substances. After 17 to 8 days, he just combined those substances and condensed by using the condenser and collected. What he collect, they got a bath. And from that day, they start supporting the Oparin-Haldane's theory, because the Oparin-Haldane theory was actually proved by Miller and Urey. What they got certain soup-like substances. That means not the plain water, but water with certain substances, chemical substances, which were identified as amino acid structure. Okay, those amino acids are the basis of this RNA in that time. That's why it was considered as RNA in primitive life were formed fast. Okay, the synthesis of critical biochemicals in absence of chemicals formed by living system, possibly what would have occurred before life was present on Earth. So most commonly cited evidence for abiogenesis in science are playing important role. That idea was provided by Miller and Urey. It was prebiotic chemistry. Okay. And at last, transition to DNA and protein. From RNA to DNA, RNA is less stable than DNA, making DNA a more reliable molecule for long-term genetic storage. Why this happened? Because early life was the simpler life, but present life is the complex life. When simple to complex occurs, stability is more important thing. Otherwise, these complex substances unable to survive. That's why RNA in comparison to DNA was not stable or more unstable in comparison to DNA. That's why RNA in turn converted to DNA for its complexity as well as for its stability. Over time, DNA likely evolved from RNA to take over the role of genetic storage. due to its greater stability and accuracy in replication. This is another important factor in replication point of view. DNA can form more corrected replication form in comparison to RNA. There is more chance of mutation, more chance of error in RNA replication in comparison to DNA. Okay, and from this RNA to protein, because proteins are more versatile catalysts than ribozymes. Whatever substances present in the primitive life. As life evolved, proteins took over most of the catalytic role, with RNA retaining its role as an intermediary between DNA and protein. If we are talking about central dogma theory, the central dogma theory explains what? Explains RNA is the intermediate product, but RNA is not the deciding product now. DNA actually decides. DNA form intermediate product, that is RNA. Then this RNA is responsible for the protein production. That central dogma theory nowadays explains. Okay, so these are the factors support RNA world hypothesis. It stated simply what? The primitive life, the simple life, they didn't have DNA, they have RNA. That's why we can say that life originated on Earth based on presence of RNA, because when protocells were formed, why they are called as protocells? Because they do not divide, they do not form the new cells, they only form from the abiotic substances, but these cells are not reproducing new cells. But when RNA was formed inside them, they start reproducing, and complexity was developed from that time. Okay, that is RNA world hypothesis. Okay, all these supports already we have discussed in previous slide. This is all about the first unit of this chapter. Am I audible?
Yes, sir.
Yes, sir.
Is everything visible to you?
Yes, sir.
Sir.
Okay, any doubt from any part? You can ask me one or two questions. We can decide, we can discuss. From any part, you can ask any questions. Yes, Sunil Naik. Any questions you have? Mr. Sunil Naik, I think you have some doubt. If any question, you can ask me.
ଆକ୍ଚୁଆଲି ସାର୍ ଏବେ ଯୋଉ ଡିସ୍କସନ୍ ହେଲା ଆମକୁ କିଛି ଡାଉଟ୍ ନାହିଁ, ବଟ୍ ପଢିଲା ବେଳେ ଯଦି ଡାଉଟ୍ କିଛି ଆସିବ ଆମେ ଆପଣଙ୍କୁ ଜଣେଇବୁ।
ହଁ, ମୁଁ ସେଟା ସବୁବେଳେ କହିଛି ଯେ ଏଇ ଯୋଉ ଷ୍ଟିପୁଲେଟେଡ୍ ଟାଇମ୍ ଭିତରେ ଟିକେ ସବୁ ଜିନିଷ କଭର କରିବା ଡିଫିକଲ୍ଟ ହଉଛି, କାରଣ ଆମକୁ ବହୁତ କମ ସମୟରେ ବହୁତ ଜିନିଷ କଭର କରିବାର ଅଛି, କୋର୍ସକୁ ଶେଷ କରିବାର ଅଛି। ତ ଏନି କାଇଣ୍ଡ ଅଫ ଡାଉଟ୍ ମତେ ପର୍ସନାଲି ପିନ୍ କରିପାରିବ ହ୍ୱାଟ୍ସଆପ ରେ, କି ଫୋନ ବି କରିପାରିବ। ମୁଁ ଯଦି ଫ୍ରି ଥିବି ସେଇ ସମୟରେ, ନାହିଁ ତ ମୋ ଟାଇମରେ ମୁଁ ତମକୁ ଡାଉଟ୍ କ୍ଲିୟର କରିପାରିବି।
ୟେସ୍ ସାର୍, ଥ୍ୟାଙ୍କ ୟୁ
ସେଥିରେ କିଛି
ସାର୍।
ହେଜିଟେସନ ନାହିଁ।
Thank you,
Thank
sir.
you, sir.
Any kind you are free to discuss with me regarding your topic, regarding your doubts.
Thank you, sir.
Okay. So, shall we wind up the meeting here?
Yes, sir.
Yes, sir.
Yes.
Very good night to all.
Good night, sir.
Thank you, sir. Good night.
Thank you, sir.